So in preparation for Lab 5, this video is going to review some basic concepts related to fMRI. So you may be already familiar with some of these concepts from the textbook, but I thought it'd be helpful to review some of these concepts. So let's start with what is fMRI. So fMRI stands for Functional Magnetic Resonance Imaging. Essentially, we're using a strong magnetic field to look at changes in blood flow in the brain. And so this is a bit different from a structural MRI, which just looks at anatomical features like gray and white matter. And this is a picture of the scanner that is actually used in research studies at CU Boulder. So what does fMRI actually measure? fMRI measures bold signal, so it's essentially blood oxygen level dependent signal. So when areas of the brain use more energy, this results in a flow of oxygenated blood to that area. So oxyhemoglobin, which has these oxygen molecules attached to it, versus deoxyhemoglobin without the oxygen molecules, these have different magnetic properties, and that allows them to have different MRI signals. So the bright areas in an fMRI scan are areas with a higher concentration of oxyhemoglobin relative to deoxyhemoglobin, and we interpret that as those brain regions being more active. So how does fMRI analysis work? So essentially we start with our fMRI scan data, and you can think of this as essentially a 3D movie. We can analyze it in different ways, but one way is to start by looking at voxels. So a voxel is essentially a 3D pixel. This is the smallest unit of measurement in an fMRI scan. So we're just looking at this little cube of brain activity over time. And if we focus on this single voxel, we can map out its signal over time. And if you look at this pattern, you might see there's some predictability to it. So there's these peaks that happen, and right after there's these valleys. So one possibility is that this regularity might be explained by different task conditions. So maybe during the peaks, they're experiencing task condition A, maybe they're memorizing word pairs, for example, and maybe B is a different condition like the rest period in between trials. So if we average this pattern over time, we might get a bar graph like this, where we can see that this voxel shows higher signal amplitude for condition A compared to condition B. Then we can do this for all the voxels in the brain and create this map that shows which voxels, which areas in the brain are more responsive to task A versus task B. Another concept I want to review is correcting for multiple comparisons. So I'm going to start by giving you an example where we talk about coin flips. So let's say we're testing for magical coins. I have a coin and I want to test if it is a magical coin that is biased towards tails. And so the way I'm going to test this is I'm going to flip it 10 times in a row. And if this is actually just a regular coin, then it would be very unusual to get 9 or more tails out of 10. So that would be less than a 5% chance, or in other words, a p less than 0.05. And so if this happens, I'll assume that I actually have a magical coin. But what if I have 100 coins I want to test? Do you think the chances of one of these coins ending up with 9 or more tails out of 10 will increase just intuitively? So yes, if I try this experiment with 100 coins, I would actually expect 5 of those experiments to end up with 9 or more tails out of 10. Again, it's just 5% of those trials. So since I was testing so many coins, my chances of getting 9 or more tails out of 10 increased, but I don't actually want to assume that those 5 coins out of 100 are magical, because now that I've thought about it, it actually seems like those were just due to chance. So in other words, this means that our standard p less than 0.05 doesn't really work. And so we need to correct for the fact that we're doing a lot of different tests. And so there are a couple different ways to do this. If you're looking through fMRI papers, you might see them mention things like FDR or FWE correction. And at this point, it's not so important to worry about how these corrections work as long as you understand why we use these, which is because we need to correct for the fact that we're doing a lot of different tests. So how do these coin flips relate to brain activity? Well, brains are a little bit more complicated, but you can actually think of each brain voxel like one coin flip experiment. So in both cases, we're measuring a process over time and seeing if the pattern is different from what we'd expect by chance. But since we're doing this for 100,000 voxels or so, we definitely need to correct for false positives.